Hi, my name is Pamela Coons, Associate Professor of Medicine in the Division of Oncology at Yale School of Medicine and Yale Cancer Center. I'm excited to announce ASCO's new open access journal, JCO Oncology Advances. As the inaugural editor-in-chief, I hope to support JCO Oncology Advances to become the premier platform to bridge the gap between accessible scientific research and clinical care. Stay tuned for more information, including new article types, at ascopubs.org/JCO Oncology Advances. We look forward to seeing your submissions in spring of 2024.
This JCO podcast provides observations and commentary on the JCO articles, a Phase 2 study of the MEK inhibitor selumetinib in patients with advanced hepatocellular carcinoma by Bert O'Neill et al., and a multi-institutional Phase 2 study of selumetinib in patients with metastatic biliary cancers by Tanios Bekesab et al. My name is Alan Vanook, and I'm a professor of clinical medicine at the University of California, San Francisco. My oncologic specialty is gastrointestinal malignancies. The podcast is entitled MEK Inhibitors in Hepatobiliary Cancers. The description: the term targeted therapy is often used in cancer drug development. This podcast will discuss the study of a MEK inhibitor where the investigators actually looked at the impact on the target as well as whether the drug worked or did not work. I wish that I could begin this discussion by announcing that selumetinib is a spectacularly effective targeted therapy in hepatobiliary cancers. But unfortunately, that is not the case. The monotherapy activity of selumetinib was modest and only observed in biliary cancers. What makes these two papers noteworthy is the pathway being targeted, called MEK, and the correlative science done with tumor biopsies to assess the adequacy of target inhibition. That these are rare diseases where tissue acquisition is often difficult makes the successful conduct of these trials even more special. First, the acronym. What is MEK, other than the target of numerous inhibitors in drug development? MEK, the middleman in the RAF-MEK-ERK pathway, stands for mitogen-activated protein kinase 1. This signaling pathway is central to numerous cellular processes that can go awry and lead to cancer, with phosphorylated ERK ultimately translocating to the nucleus and causing unregulated growth. The cascade is activated in many cancers through mutations either within the complex or upstream, such as RAS or EGFR. Inhibition of MEK has been effective in controlling tumor growth in numerous preclinical model systems, placing MEK, like EGFR or VEGF, in the crosshairs of targeted therapy strategies. Selumetinib is one of numerous inhibitors of MEK. In fact, at last count, at least 10 drugs that are purported to block this pathway are in clinical trials. An orally available small molecule, selumetinib is thought to be more potent and selective than the other molecules in its class, at least in experimental models. The mutations of interest are believed to be common in cancers of the biliary tract, but are uncommon in hepatocellular carcinoma. However, the efficacy of sorafenib, perhaps a RAF inhibitor, in hepatocellular carcinoma supports the potential significance of the pathway. Even with a rationale for testing this class of drugs in these patients, it goes against conventional wisdom for an agent to be tested in these cancers early in its development. In fact, until the success of sorafenib in hepatocellular carcinoma, companies and investigators alike shied away from these diseases entirely. Sick patients with liver disease as a competing cause of death and impaired hepatic function, making the metabolism of the drug and the toxicity unpredictable. What's more, the ability to visualize and measure these tumors is often compromised, presenting a distinct disadvantage to early drug development. So we must compliment the leaders of the two studies, Bert O'Neill and Tony Bekesab, for boldly tackling these diseases. But the effort went beyond the straight clinical question of tumor response. Each study required tumor tissue for analysis, and the hepatocellular cancer study actually required biopsies before treatment and during treatment, although cost constraints limited the paired sampling to just 5 patients. The analysis of before and after samples in the liver cancer study allowed the investigators to determine whether the target was actually hit. In fact, the finding of a decrease in phospho-ERK in 4 out of 5 patients suggests that the agent does what it is supposed to do, inhibit MEK. Unfortunately, the inhibition of MEK did not mean tumor shrinkage, of which there was none in the hepatocellular carcinoma patients. However, there was also no ocular toxicity seen. Retinal complications with other MEK inhibitors are so common that they are felt to be an on-target effect, and patients enrolling on trials with MEK inhibitors are followed by ophthalmologists. Therefore, while a decrease in phospho-ERK was noted, it may be that the degree or duration of MEK inhibition was still suboptimal, leading to no anti-tumor or clinical retinal effects. In the biliary study, a systematic characterization of the genetic mutations of each patient's cancer was conducted. A modest response rate in the biliary patients suggests there may be some potential for the agent in those diseases, albeit without an obvious laboratory correlate to follow. These studies were challenging to patients and investigators alike. They exemplified the design and execution of informative Phase 2 trials. While we learned that there may be some activity of this MEK inhibitor in biliary cancers that is worth pursuing, we also learned that our surrogate measures for MEK inhibition may not be predictable, even if measurable. It is such science that guides the development of future trials and may someday lead us to the treatment paradigms that justify the term targeted personalized therapy. This concludes this JCO podcast. Thank you for listening.
For more original research, editorials, and review articles, please visit us online at jco.org. This production is copyrighted to the American Society of Clinical Oncology. Thank you for listening.